हॅलो.
आपको बोला जाएगा तभी बोल बोलता हूँ मैं सर यार बोलत नाही काय खरंच काय बोलत नाही मला कुठं जावं नाही. मला विचारते तेव्हा नाही बोलत.
Today's guest speaker, Dr. Vibin sir has joined this conference. I welcome him to the conference. Welcome, sir.
Thank you so much. Thank you.
We will begin with our conference very soon, just within 10 minutes, sir. Should we begin with the program, sir?
Sure.
Good morning to everyone, and it's me, Vaishali Chaudhary, welcoming you again on the second day of International Conference on Recent Advances in Chemical and Biological Sciences, organized by Gokhale Education Society's NB Mehta Science College Bordi. It was a fruitful, enlightening day yesterday where we had the privilege to listen to distinguished speakers, Professor Satya and Professor Vivek. We had some technical issues in yesterday's session, but I really appreciate the cooperation given by you all. Thank you.
Good morning to one and all. So let us begin with the session. Now I request Dr. Kirti Kulkarni Madam to introduce invited speaker, Professor Vipin Ramakrishnan Sir, IIT Guwahati. Over to you, Madam.
Thank you, Benazir Madam. Good morning to all of you. Respected Principal Madam, dignitaries of the session, delegates, my colleagues and participants, myself Dr. Kirti Kulkarni from NB Mehta College. It's my privilege to introduce today's invited speaker, Honorable Dr. Vibin Ramakrishnan. Dr. Vibin Ramakrishnan is a professor of biosciences and bioengineering, IIT Guwahati. Dr. Vibin earned his master's in applied chemistry from Cochin University of Science and Technology and PhD from School of Biosciences and Bioengineering, IIT Bombay. Sir worked as a postdoctoral research associate at Rensselaer Polytechnic Institute, New York, and a faculty scientist at IVAB Bangalore before joining Department of Biosciences and Bioengineering at IIT Guwahati. He has about 50 publications in reputed international journals like American Chemical Society and Royal Society of Chemistry. and also 10 patents, including an international patent to his credit. Sir is the principal scientific advisor of two biotech companies. Vipin Sir is the editorial board member of two reputed journals, Journal of Controlled Release of Elsewhere and Scientific Reports of Nature Publishing Group. The Department of Biotechnology, Government of India has awarded Sir the Innovative Young Biotechnologist Award IYBA in the year 2007 and 8 to recognize his contribution and innovative approach in biotechnology research. Sir's interests are network medicine, drug delivery vehicle, and bio-nanocatalysis. Sir has achieved Innovative Young Biotechnologist Award IYBA 2007 and 8 from Department of Biotechnology, Ministry of Science and Technology from Government of India and its extension in 2011 and 12. Also, he achieved postdoctoral fellowship funded by National Sciences Foundation, United States. Sir is a member of National Academy of Sciences India, member American Chemical Society. He is also member of International Society for Computational Biology and also Indian Biophysical Society. Sir, we are very eager to listen to you. I request you to kindly deliver your talk. Over to you, sir.
Uh, thank you so much. Thank you, Professor Kulkarni. Thank you, Professor State.
Thank you.
Thank you, Principal and Administrators of NB Mehta Science College. Thank you, Kirti Patel, for inviting me. Thanks a lot. So, can I share my presentation so that please let me know whether.
Yes, sir.
Can you see the slides?
Yes, Vipin, it's available. It's visible. Sure.
Okay, all right. Yeah. Thank you so much. Thank you so much. All right. So good morning to you all. Good morning, students of NB Mehta Science College and all other student community who have joined for this conference. It's a pleasure to join the student community wherever it is and whichever part of the world, and I appreciate that. And I'm thankful to the organizers once again. In this lecture, I would like to talk about the reductionist approach to molecular design and activity profiling. I'm Vipin Ramakrishnan. I'm a professor of bioscience and bioengineering at IIT Bordi. And the very idea of this comes in the very next slide. Many of you must have already heard about the concept of reductionism. Concept of reductionism, actually, when we see the literature, it dates back to, say, 650 BC. And after that, many philosophers, especially Greek philosophers like Socrates and Plato, have been proponents of this philosophy in one way or the other. There are different variants of this philosophy of reductionism. From an Indian perspective, the Sankhya system of philosophy, or Sankhya Shastra, we call it, by sage Kabila Maharshi, there is a first mention of this reductionist approach. This is to the best of my understanding. It may not be correct though. So the basic idea, how I conceive it, is that how do you reduce information? The beautiful way of reducing information, or this classic example, is that you can reduce a tree to a seed. All the informations that are required for a tree to grow in its full-fledged size and shape is actually encoded in a seed. And all its functions also are in one way or the other encoded in the seed. So there is a beautiful way that nature encodes its information in an encapsulated format, is it not? An extended version of the same idea is there in Indian philosophy, that the whole world exists in a seed state, just as a tree exists in a seed in a causal form. Whole universe exists in an inverted form, Urdha Mula Madoshaka, if I remember it correctly. All right. So this is the basic idea of the concept of reductionism. Now our story is, actually we have two stories to tell you in about 30 minutes. They are interconnected stories. The first story is of reducing molecules, small molecules, to time sequences. And the second story is about reducing the molecular size of fairly large molecules, or fairly large functional molecules, to a very minimal size that is just required to perform its function. So I will explain this in next 30 minutes or so. All right. To start my presentation, I would like to invite your attention about my personal fascination with a concept called time. So basically, this is a brief history of my fascination with time. When I was a student, I asked my teacher that how much is 1 hour, and obviously the answer was 60 minutes. And the next question is how much is 1 minute, and the answer is 6 seconds. And when I asked how much is 1 second, my teacher did not like it actually. And the answer to that question, I had to wait till class 11. And in NCERT textbook of class 11, you can see a statement that 1 second is the time needed for a cesium-133 atom to perform 91926317770 complete unperturbed internal oscillations between two hyperfine levels in its ground state. So this is the standard definition of a second. Okay, all right. But the time in its philosophical existence is much more than this, you see. Everything that which we come across, almost all biological systems, almost all material systems, it's a function of time. Its evolution is a function of time. Or everything that is evolving is latently a function of time. Something that is so microscopic or so small, defined by the postulates of quantum mechanics, or something that is fairly large, defined by the postulates of Newtonian mechanics. There is a time factor. There is T in almost every equation, isn't it? Even this Professor Stephen Hawking's book is a brief history of time. You see, there's time. And if you think about time, when you read philosophical books, like for instance, Bhagavad Gita, you will see the statement that I am devoid of matter, space, and time. So what he says is that time is not a function of my existence. If we think about it philosophically, what is time? If anything, can anything be devoid of time? Can anything be not a function of time? And the only thing that which comes to my mind is anything that is not a function of time, then it has to be the time itself. You see, time itself is not a function of time. Okay, so I got a corroboration of this idea from Bhagavad Gita itself, in saying that kalosmi lokatraya pravarto. Kalosmi means I am time. So one has to be a time if he has to claim that I am devoid of matter, space, and time. Right. Now time, its time is very, very interesting. Time can be used to represent other dimensions also. When we were students, if you ask, like from our campus, how long is CST railway station? But we used to say it's just about 50 minutes. You see. 50 minutes, but the question is how long or what is the distance between your campus to CST railway station? But the answer is in minutes, because it takes from Kanjurmarg station to CST, it's about 50 minutes by local train. And curiously, you see, light year is a measure of distance. So time is actually used to represent other dimensions also, and very freely used to represent other dimensions also. So time is everywhere actually. So what we thought is that because time has an omnipresence, can time be also made use of to represent molecules or predict the activity of a molecule? So this question is the crux of the first part of this presentation. And why we are doing it, how we are doing it, and for what purpose we are doing it is the subject matter of next 10 minutes. Now, let me start with the question why. The why question is because the reason why we are doing this, we researchers, when we embark into any new research problem, we always make sure that we are doing it for a purpose, because otherwise there is no value for it. So we are doing this particular project also for a particular purpose, because the discovery of new chemical entities is much less than the expectation of industry. This is a phenomenon that is especially very relevant in this millennium, after 2000. And one possible solution to this is the molecule is to optimize the molecule as much as possible in the early stages of pipeline, because later stages of pipeline is very costly. If they enter into clinical trials and if it is not approved, then the company or the institution will lose billions of dollars, and it will be very difficult for that company to cover that cost at a later time. So the strategy that is adopted is to optimize a molecule as much as possible in the early stages of a pipeline. And there is that is the opportunity where an informatician fitted, whether it is somebody from bioinformatics area or somebody from chemoinformatics. Okay, so that is the purpose of it. We will try to see, visualize, and assess the quality and efficiency and utility of a molecule as much as possible in a computing machine. There may be a difference of opinion to many people, but I always say to my students that a computer is the most versatile experimental device. I repeat, experimental device ever discovered in the history of science. All right. Now, let me start with how we are doing it. The first question is why we are doing it. Now we are saying about how we are doing it. So this is the project. The idea is to create a molecule that preferentially binds to a protein target whose structure may or may not be well characterized. If you see the structure on the right side, it's acetyl salicylic acid. You are all chemists and you know it. Aspirin, commonly known as aspirin. Now our idea is to do a virtual activity profiling of this molecule against a fairly large database of millions of molecules. So how do we do this? So this is the whole idea. The challenge is that if you have a hypothesis or a structure, then you should be able to search a fairly large database of millions of molecules, screen it out to a few hits, and that how to do this particular exercise, the algorithm that which we are proposing to do this exercise, and the efficiency of that algorithm and that method, what we call as clock hypothesis or a clock model, is the subject matter of this discussion. So the challenge here is to get one or few answers from this huge heap of possibilities. All right. For this, if you want to do this, then you have to do something called canonicalization exercise. Canonicalization is actually a computer science concept. For instance, for chemists to understand Fourier transform is kind of a canonicalization exercise. The idea is, if of this canonicalization exercise, to quantify the necessary elements or properties that which we need and convert it into a mathematically amenable or computationally amenable representation. For example, if you want to compare between a crow and a pigeon, or a pigeon and an eagle, for instance, we know that by seeing it, pigeon is much inferior in its ability to fly than a pigeon. Okay, pigeon can fly at longer distances. It can be airborne. It can be airborne for a longer time. But just by observing it, we understand it by observing it. But this observation is of no use when we are comparing with millions of molecules. We need to convert it into a computationally amenable representation. So I'm trying to say an example for you that I'm defining a term called quantitative word structure property relationship. For instance, QBSPR, I'm calling it, and I'm evaluating this performance as muscle efficiency multiplied by the flight time, right? So you will get a number for pigeon and you will get a number for eagle as well. So by comparing these numbers, you can compare the efficiency of their performance. So then when it becomes a number, then it comes to a computationally amenable representation. So similarly, we should be able to represent a structure, a molecular structure on towards your right, to a computationally amenable representation if you want to compare it to millions of structures, right? This is the first such attempt by David Binninger in 1980. This is canonicalization of a chemical data. This is called Smiles representation. If you can see it, that either alcohol, CH3, CH2O, it is represented as CCO. And there are many such string representations for various molecules. The idea is that this molecule, which is in 2 or 3 dimensions, is translated to a 1-dimensional string representation. All right. Now, how do we do it? What is our approach in doing it? Our approach is the following. I'm taking the same example as acetyl salicylic acid. We have the following functional groups in this. We have a benzene ring, we have a carboxylic acid group, we have a CH3, we have a CO. So we identify these functional groups as pharmacophoric elements. or PPPs, potential pharmacophoric points. That they are called PPPs. We define it as PPPs. And these potential pharmacophoric points, we enumerate these potential pharmacophoric points. Now imagine a situation that I have this molecule acetyl salicylic acid. I'm keeping a big model of this molecule in a closed room, okay, which doesn't have windows and probably doors. Now what happens? If I shine light on it, then I can get an image of this molecule on the wall of that particular room, is it not? So I can reflect this molecule on the wall of a room. Okay, that is the first step. Now I have a reflection of this molecule on the wall of a room. This is my molecule. Now on this reflection, what I do is that I assume that this reflection is imprinted in a clock. Suppose that particular wall I have an image or a background representation of a clock. What it offers me is that I can now tell you, once I reflect that I have a functional group like COH at, suppose this is the image, and I can say I have a COH at around, say, at around 1 o'clock. I have an ether, it's not ether, sorry. I have a carbonyl group at around, say, 4 o'clock. I have a CH3 group at around 3 o'clock, and so on and so forth. This is not. So what it offers me is my three-dimensional structure is now represented in a clock, which is in 2 dimensions. And then from an analog clock to a digital clock or a digital watch, you can always convert it. What I will get is a string representation. This string representation is very much like what you see it in a digital watch. So here in this case, you can see at 0, there is A, at another 0, there is G, 0, 7, there is D. What does this mean? To get an answer, you can see the right side of this particular slide. I have defined a series of functional groups, and I have given alphabets to denote these functional groups. So at 6 o'clock there is Z, means 6 o'clock there is a halogen. I mean, there is a fluorine, chlorine, or bromine, whatever. That is what it actually means. And what it offers you is that you can reduce this molecule, which is in 3 dimensions, to a single dimensional string. And this single dimensional string encodes as much as possible. I am not saying it is a perfect 3D, but encodes all 3-dimensional information that is required to try to define a molecule. Okay. Now, if you reduce this 3D to 2D to 1D, what we get is an interact. You can also get an interaction fingerprint with a molecule to a target as well. What you essentially get is a 1-dimensional representation as a time sequence. You will get a time sequence. Okay. Now, what is the advantage of this time sequence? I have a very selfish motive to convert this into a time sequence. Is because you have a series of sequence comparison tools and algorithms already available for comparing amino acid sequences and nucleic acid sequences. I can reprogram it without much difficulty, use the same algorithms and tools for my time sequences as well. So if amino acid sequence is in the bioinformatics space, I have a time sequence in the chemoinformatics space. And if you don't believe it yet, then these are 3 famous receptors, which are actually drug targets as well. And what you can see on the right-hand side are their natural ligands and those drug molecules or candidate drug molecules and how they align against each other. You can see the same letters being repeated at specific time points. So 0 to 11 is the specific time points, and I can easily convert this to a 1-dimensional string representation. And this is how this particular algorithm, this particular model fares against the standard established systems, which does similar kind of work. You cannot have 2 models with this exactly the same idea. So what is the summary? You see, suppose a student in NB Mehta Science College is joining a company in Bombay, for instance, and his principal investigator in that company, he can say that, look, you have to make a molecule, synthesize or design a molecule which has a hydrogen bond donor at 3 o'clock and an acceptor at 7 o'clock and 10 o'clock. Then he should be able to write that time sequence and do a reverse engineering to get into the molecule. So this is the idea. This idea is actually published in Molecular Informatics in roughly about 2010 in collaboration with Andreas Bender of University of Cambridge Chemistry Department and my earlier student Amy Sebastian, who is now working in Los Alamos National Laboratory, is the person who have written this code as well. Okay, that is the first part of my story, and I think I have 10 more minutes. The second part of my story is the story of a blue fluorescent protein. It's a story about reducing the molecular size of a functional molecule to a very small entity of, say, one-tenth of its size, roughly. Okay. So let's go with the story. So first, we have completed a story of a small molecule. Let's see how the idea of reductionism can be also employed in a larger molecule. You all must have heard about this molecule, green fluorescent protein. It is a very important molecule, at least in biotechnology space. Martin Schalfie, Roger Sine, etc., have got Nobel Prize in 2010 for the discovery of this molecule. This molecule is very curious. It's a very important molecule, actually, because it is actually first sort of extracted from jellyfish. And this molecule has an excitation peak wavelength at around 395 nanometer, and it emits at 509 nanometer. So that because it is emitting at 509, it is blue fluorescent. It is naturally, sorry, it is green fluorescent. It is naturally green fluorescent. That's why it is called a green fluorescent protein. This protein has about 238 amino acids. And the fluorescence is a result of fusion of 3 amino acids, serine, tyrosine, and glycine. And this green fluorescent protein exhibits a very bright fluorescence. What I thought is that, can we actually make a protein? This protein is about 238 amino acids. Can we make a protein of 25 or 30 amino acid long? Means idea of reductionism. Can you reduce it to the essential parts, to the most important aspect of it, and then make a molecule which has which does similar work, which is fluorescent, basically. Okay. These are the computational algorithms and tools that are used. One is ARSAMD, Automated Repetitive Simulated Anything Molecular Dynamics, and the other one is IDEAS. Both these tools are being used in the design phase of these molecules. The details of these tools are published in two separate physical biology papers in the same year, 2018. Your colleague or your teacher, Dr. Kirti Patel, is an author of both those publications, so you can ask him the details. So I'm not going into the details. Instead, I will restrict my discussion in the storyline. So we could design about and synthesize about 5 sequences, and of which the most efficient one is the third one, BP02. So we have made an amino acid sequence of about 30 amino acids, which has both L and D chiral amino acids. So it is a heterochiral amino acid sequence. And in the core of this, we have azulinylalanine, a molecule called azulinylalanine, which is an artificial amino acid. This azulinylalanine will excite at 348 nanometer, and it emits at 390, 400, which makes it roughly violet indigo blue, or kind of a bluish color. That's why it is a blue fluorescent. It can potentially be a blue fluorescent protein. And this particular protein is kind of pregnant with this azulinylalanine unnatural amino acid. And when you have a water exclusion, this protein will be blue fluorescent. So this is the excitation wavelength and emission wavelength of this particular protein on the right. And this is actually how it looks like in a microcentrifuge tube. So BP02 is beautifully fluorescent. And this is arguably the smallest artificial blue fluorescent protein ever discovered in the history of science. And it is the first heterochiral blue fluorescent protein, ever discovered in the history of science. It is published in American Chemical Society's Omega. So thank you so much for listening to me. This is my research group, Molecular Informatics and Design Laboratory. This is a picture of IIT Guwahati, an aerial picture of IIT Guwahati. We are on the banks of Brahmaputra, as you can see it on the rear side. And these are my present PhD students, and these are graduated PhD students. Most of the work that I have described in the later part is done by Vivek Prakash. And in the first part was done by Amy Sebastian. She is in Los Alamos National Laboratory and in collaboration with the Andreas Center of University of Cambridge. Thank you so much for listening to me. Thanks a lot. This is my contact email address. If you have any questions or any further queries on this, please don't hesitate to contact me. Thank you so much. Thanks for this opportunity. Once again, thank you for the organizers for inviting me.
Thank you, sir. Thank you so much. Is there any question?
I have a question. This is Roop Malik from IIT Bombay. Can I go ahead or I should?
Yeah, sure. Thank you. Thank you.
Hello, Professor Vipin. This is about the last part about making an artificial fluorescent protein. That's really impressive. But I was just wondering because if you think about green fluorescent protein, I mean, a lot of that peptide is used in making the barrel cage around the chromophore, right?
Correct.
So if you if you have protons, if you don't have that cage, you will have proton exchange with the internal chromophore.
Correct.
You don't get much fluorescence. So I am really surprised how you can, make this peptide so much smaller, presumably without the external barrel, and still get fluorescence.
Yeah, okay, all right. So that is basically the subject matter of the third slide or fourth slide from the last, because we use two computational tools, AR simulated annealing molecular dynamic simulation. And what we do is that we take about a 30 amino acid long polyleucine, all right, and we heat it up to 800 Kelvin, and we take off the functions that makes them remaining in a particular stereochemistry, polyol stereochemistry, and we allow the molecule to anneal. And when the molecule coming down from 800 Kelvin to 300 Kelvin, the molecule will form a very compact structure. Okay. And now after that, we change this polyleucine sequence to an amino acid sequence by using a tool called IDEAS. That is an automated protein design tool which we have developed in IIT Bombay when I was a student over there. And this gives me a sequence to that. And I purposely allow a sequence of tryptophan in the core of that protein. Now, in the next step, I mutate that tryptophan to azurenyl alanine because tryptophan actually excites at around 280 and it emits 340, 345. That won't give you a color actually. So I wanted a colored protein. It sounds like a fancy project though, but it is indeed a fancy project.
So the fluorescence excitation and emission spectra you showed are computational spectra? They are real?
No, they're real experimental spectra.
They're real experimental.
Real molecules. We have synthesized this molecule by solid phase peptide synthesis.
Okay.
And the BP02 molecule is quite stable also.
And what is the quantum yield like?
I'm not very sure about it.
Okay, sure.
I'm not sure about it. I'm sorry.
Thanks.
I don't remember it actually. In fact.
Okay.
Yes, any more question? Yes, I think there is no more question, sir. Thank you, sir, for enriching our knowledge in the field of reductionist approach to molecular design. Sir, you have given very beautiful example of seed and tree to explain this difficult concept. Sir also explained the concept of time as a function with the example of Gita and Sankhya Shastra. Sir has answered many WH questions regarding drug designing and protein molecules. Sir, your thought-provoking ideas are really source of inspiration for all of us. On behalf of the college, I express my sincere thanks to you for sparing your valuable time with us. Thank you so much, sir.
Thank you so much, and thanks for this opportunity. I'm very happy to talk to Professor Roop Malik after a long time through this platform. Thanks a lot.
Thank you, sir.
Thank you, Kirti Madam. Our next invited speaker is Professor Roop Malik sir, who is closely associated with our college and is very keen on development of the college. I welcome you, sir, for the conference, and I request Ms. Vrishti Patel from Department of Biotech to introduce Professor Malik. Over to you, Vrishti.
Thank you, ma'am. Good morning, one and all. Myself, Vrishti Patel, from Mehta Science College, Bordi, is here to introduce Professor Dr. Roop Malik sir. It gives me immense pleasure to introduce a personality who is a perfect blend of all the branches of science. By his multidisciplinary approach, he has truly exemplified the fact that knowledge knows no bounds. Sir is currently a professor at the Department of Biosciences and Bioengineering, IIT Bombay. His lab is working to understand how motor proteins help in degradation and clearance of pathogens, and also how these motors work inside the liver to maintain systemic lipid homeostasis in the animal. Throwing light on his academic background, Sir has completed his master's in physics from University of Allahabad, Uttar Pradesh, PhD in physics from Tata Institute of Fundamental Research, Mumbai. Dr. Roop Malik Sir has been a postdoctoral fellow at TIFR Mumbai and National Center for Biological Sciences, Bangalore. Also at University of California, Irvine, Department of Developmental and Cell Biology. He has also worked as Senior Fellow at Reader and Wellcome Trust UK and as I Senior Fellow at Associate Professor and Wellcome DBT Professor and DBT. So such a proficient and versatile personality is recipient of many awards. So to mention few, Sir is the recipient of Shanti Swarup Bhatnagar Award in Biological Sciences 2014, Infosys Prize in Life Sciences 2018, GD Birla Award for Scientific Research 2019, Wellcome Trust DBT India Alliance Senior Researchship Fellow, Wellcome Trust UK International Senior Research Fellowship, Human Frontier Sciences Program, Long-Term Postdoctoral Fellowship. So out of many publications, I would like to give some glimpses of his important publications. He has published From Physics to Physiology at the Membrane Motor Interface, Insulin activates intracellular transport of lipid droplets to release triglycerides from the liver. Coin tossing explains the activity of opposing microtubule motors on phagosomes. Kinesin-dependent mechanism for controlling triglyceride secretion from the
May I
liver.
request you, that's good enough. It's okay. When I am writing my CV next time, I will contact you. Okay?
Okay, that's so kind of you.
Thank you. You did not have said so.
Okay, I would like to hand over the podium to Sir, and I would request you to enlighten us with your knowledge.
Yeah, thank you. Thank you, Roshni. Let me just share my screen before we start. Can you see anything? You can, I guess, right?
Yes, sir. It is visible, sir.
Yes, sir.
And it is not mirroring or anything like that, right? Now full screen?
No, sir. But sir, it's not in the full slide mode. Sir.
Now it must have gone to full screen. Now?
No, sir. It is still not in slideshow mode. Sir, you will have to share the slideshow mode window.
No, I did that. I think I have to share my entire screen, otherwise it doesn't work.
Professor, it will help if you share the entire screen. Yeah.
Yeah, I'm doing that. Looks fine. Now is it better?
Yes, sir. Now it's fine.
Okay, good. The only problem with this is that I can't see anybody in front of me. But hello everybody, and good morning, and thank you Vrishti and Vaishali and all the other my friends at Bordi College. As many of you know, I visit you sometimes whenever I'm around in that area, and it's always a delight to come there, and some of my lab members have also visited you. And I'm very happy that you are conducting this meeting, although I think that in future, maybe sometime you should conduct these meetings in person, and I would very much like to be there when you do so, okay? Yeah, so I have about 25 minutes, is that correct?
Yes, sir.
Yeah, okay. So we work in the general area of trying to understand how motion happens, how in biology, in the biological sciences, in your body, how force and motion are generated. And I'm sure all of you have heard the word, have heard the phrase that what is life without motion. So if you don't move, you will eventually die. Even something like a living object like a plant, which we think does not move, yes, it may not jump around from here to there, but there is an incredible amount of motion inside the plant, which is required for the plant to live. I'm sure all of you understand that. So we try to understand how this kind of motion is generated at the level of molecules, and then it is amplified to much larger levels. For example, me moving my hand around, or that is something very obvious, how I can move my hand around, how I can speak, how my vocal cords can vibrate so that I can speak, how my heart can beat. So all of this is examples of motion that is generated at the level of molecules and amplified to a much larger scale, so that something visible happens or something audible happens. But that is not necessarily true for all kinds of and all kinds of motion of the body is not so visible. And we are interested in the similar process, the similar molecules that cause such motion inside the tiny cells of your body, because only when we understand how things move inside the cells of our body can we understand many basic biological processes, which are also extremely important for understanding how many disease relevant phenomena. And if you look at this picture on my, so if you look at this picture, the molecules that I'm talking about are shown here in red or green, and the arrows that you see in green or here in reddish color denote the direction in which these tiny molecules can generate force. And this object that you see here, this egg-shaped object, could be, let's say, a mitochondria or a virus particle or a mycobacterium or a bacteria inside that has infected your cells. And there are these tiny force-generating molecules which bind to so many kinds of things inside your bodies. And after they bind, they generate force in this direction, which is shown. And this force, these actually move along these kind of tracks inside the cells, inside, let's say, the neurons or many other kinds of cells in your body. And when they move around, they can take this mitochondria or this virus or this bacteria to a location inside the cell where something is going to happen after this object, let's say this mitochondria, reaches that location. For example, let's say all of you know that mitochondria is the powerhouse of your cells, okay? So these mitochondria can make something called adenosine triphosphate, which is the energy currency in your cells. And when this mitochondria is carried to a certain location, you can supply adenosine triphosphate and give energy for processes. For example, the branching of a neuron in your brain, so that when a child is growing up, these neurons have to develop and they have to branch, and these mitochondria are taken to the position where these branching can happen. So that is the kind of broad importance of what we work on. Now, one example which is here, this is a typical, this is a cell under the skin. It's called a melanocyte. It is, let's say we have this, lizard. In Hindi we call it girgitan. I don't know what we call it in Marathi. But this guy is hiding around in the bushes and suddenly it sees a cat and it has to change its color so that it looks green like the leaves around it. Otherwise it will get eaten up by the cat, okay? So then what will happen, that under the skin, so these cells that you see here, you will see a movie here, under the skin of this creature, you will have these tiny balls of ink which are transported, as you saw, in about 5 to 10, few minutes from the periphery of the cell to the center of each individual cell. And when that happens, this might change, this guy might change color, okay? So it's a very, demonstration of something that is we are all aware of, but most of us may not know how this actually happens. This happens because each of these black dots that you see, this little ball of ink, is, you can think of this little ball of ink of this object which is being carried around by these proteins, so that they come to the center of the cell and the color changes. And this is another example of what happens. I told you about mitochondria. So let's say this object was a mitochondria here. Here are in the neurons these are not cultured neurons, these are not neurons in the brain, but you can see that these mitochondria, these little fluorescent objects, are moving along the neurons. But once you have a rabies-like infection, these stop moving completely, and that is, really that it is very scary that what these kind of infections can do to you. And as you know, rabies infection, if you really get infected, most often it is fatal. Okay, so now while that is the general background, largely the way people have studied these kind of proteins that generate force and organize things inside the cell is that the picture is something like this, where you have one of these objects, these proteins. These are called motor proteins. Okay, so I will show you this. So just remember that these are called motor proteins. Okay, motor because they transport something from one place to the other, and they walk in this step-like manner. This is exactly how you or me would walk on the road. They walk in this manner, and when they are walking, they are bound to this mitochondria, or to this virus, or to this bacteria, whatever, this one, and they are bound to the lipid membrane. So all of you know that many of these vesicles or organelles inside the cell, this is what they look like. Okay, and this is what they look like, and so there are so many proteins bound to the membrane, this membrane, or this blue membrane, whichever one you want to call it. And one of those, or a few of those, could be these motor proteins, and they carry this around here along these tracks inside the cells, which are the microtubules shown here in fluorescent stain. Okay. Now, one of the things which I'm taking from the first slide, as you had seen, these tiny motors, these ones, let's say the green ones are, this is the green one, and these orange ones are another kind of motors, and they exert force in opposite directions. I'm not going too much into the mechanism by which they generate force. It's not very relevant here, but they exert force in opposite directions, shown by this, direction of the arrow. So when you have the kinesin motor active, the kinesin is derived from the word kine. Kine means to move in Greek. Dynein is derived from the word dyne, which is a unit of force again, okay, in Latin. Now, when this object, let's say this little red ball here, shown here, if it has to go towards the periphery of the cell, the kinesin motors usually dominate. And if it has to go towards the center of the cell here, the dynein motors would dominate, okay? And we understand very little about how the cell decides which kind of motor will be activated at some point of time, either the kinesin or the dynein. And because of that, a particular cargo would be carried either to the periphery of the cell if the kinesins were dominating, or they would be carried towards the center of the cell if the dyneins were dominating. So if you look at this movie, you can imagine that in this picture, because after some time everything more or less comes to the center of the cell, that the dynein motors are dominating in this war between kinesin and dynein, and that's why everything kind of accumulates at the center. Okay, right. So it is important to have some idea of the scales that we are talking about. How small are we? So, if you are walking like this, your leg here would be about a meter long, right? If you can just measure, it's roughly a meter. But the leg of dynein here, this is dynein, one leg of the dynein motor. This is this motor, okay? This one of these red things here. It is seen under an electron microscope, and it is trying to do what you would do when you were walking, and it is, so many orders of magnitude, about 7 or 8 orders of magnitude smaller than your leg. So it is very different to move around in this manner when you are such a small object, is very different from moving around when you are a 1 meter, 2 meter long object. And that is because under these conditions, when you are this kind of a nanoscale motor, this is a nanoscale motor, right? Under these conditions, the issue is that viscosity completely dominates everything, okay? So the way in which you would move would be very different from what happens in this case, but that's something that we'll not get into right now. And these are all not just something which, these are marvelous machines, of course, and we are not come close to building anything like this ourselves yet, but many people have tried. And what you will know that a few years ago, the Nobel Prize in chemistry was actually given to scientists who are working in this area of research. And here is a video of a human-made nano car, okay? And this doesn't look like much, but these are the wheels of the car, and these wheels are actually moving this car around, and you are seeing this under a special kind of electron microscope. And there are many ways in which people are trying to make these kinds of nano machines. Why are people trying to make these nano machines? Because imagine that you could package a little bit of drug in one of those like inside a mitochondria, and deliver it to the right location inside the cell, and you would be able to, perhaps one day treat diseases in a very targeted manner. For example, if you have a tumor in a particular part of your body, if you could target these motors to that tumor, and when they're carrying a drug, you have a much better chance of killing the tumor, okay? So how do people make these kinds of machines? There are many different ways. One way is to use light, okay? So these are chemical molecules. For example, each of the wheels could be part of a large molecule, and it undergoes a rotation when you shine light on it, or when you change the pH, okay? And so you can imagine when there is a conformational change shown by this arrow here, okay? Shown by this arrow here, this is the wheel, okay? It is rotating, these wheels are rotating, and then this object can move forward. It is in, the only issue is that until now, the kind of biological motors like dynein or penicillin that I was talking about, the fuel that they use is floating around inside the cell, okay? And they just catch that fuel, that the fuel is ATP, okay, adenosine triphosphate. But here we are having to supply fuel in an on and off manner by changing the pH, or by switching light on and off. Now you may all ask, why is all this useful? Is it useful at all to make these kinds of machines? As very renowned scientist David Lai said, that it is like asking a Stone Age man when he's trying to build the wheel to predict motorways. Now, I don't know if it was a Stone Age man or a woman who built the wheel, but whoever it was, if he or she was building a wheel and you ask them to predict, the highways of today, it's a little bit like that. That's where we are at this stage. Okay. Now, in the context of what we are interested in, if you go back to this picture here, okay. Here is a cell. This cell could be growing on a cover slip, or it could be in some particular organ inside your body. And imagine that a pathogen is going to come in like this laser pointer here and goes inside this cell. And once it goes inside, let's say it's, for example, the SARS coronavirus, okay, or Mycobacterium tuberculosis bacterium, that pathogen has to go inside. It has to now, depending on whether it's a virus or a bacteria, it has to multiply inside the cell. And to do that, it might need to use the resources at the center of the cell. It might to read the DNA, information in the DNA of this cell, and code for its own proteins. Once it codes for its own proteins, it can build copies of itself. So one virus getting in could make 100 viral particles inside this cell. And once those 100 viral particles are made, that virus now wants to get out, okay? It wants to get out and infect the cell next to the neighboring cell, okay? And this is very important to understand, obviously, how this kind of transport inside this happens. Where do these viruses replicate? How do they get out? And they actually do this, getting in or getting out partly by hijacking these kind of motors. So if you imagine a viral particle or a bacteria sitting on these motors and moving around, they can hijack these motors. And so to understand the basic processes by which these things happen is really important. And so now if we imagine that your screen here in black, imagine that, where my cursor is, why is my cursor not moving? Yeah, imagine that this is the boundary of a cell, okay? And if I play this, you will see that something comes in from outside. This could be a pathogen, okay? Let's say a bacteria, which comes in from outside. And what happens, as I will repeat this again, you will see that this pathogen shown here in green does a little bit of back and forth motion, okay, something like this. This is of course an animation, but just to kind of give you the idea, you see this kind of a feature of the pathogen. And ultimately, after some time, 5-10 minutes or something like that, this pathogen changes its very nature. And that's why I'm changing the color from green to yellow. And that is now, earlier it was an early phagosome shown here in green. Now it is a late phagosome because it has spent some time moving back and forth. And then what happens is that this late phagosome moves in a very different way. You saw that it just directly went here from its location straight inwards towards the nucleus. And there it fuses with these red particles called the lysosomes, and these lysosomes are acidic and they are required to kill this pathogen, okay? So many pathogens are smart and they will just sit here, or they will just sit somewhere else on this pathway and not go to the lysosome and therefore not get killed, okay? So this is one of the, so what we do to try and understand this process, one of the kind of experiments that we do is that we take cells, grow them on a cover slip, and here is one cell. This is the boundary of the cell. You are seeing part of the cell. The nucleus of the cell is somewhere here, okay? And to mimic this pathogen, we feed it with these small plastic beads, okay, 1 micron. And you can see how they move around inside the cell. This is happening in real time. They can move really fast, okay? And you can see this guy moves straight to the center, okay? And now this motion is happening. This motion that you see is not just diffusion. It is happening because you have to imagine these tiny motors are sitting on these individual beads. These are 1 micron inside. They're very small beads and they're carrying them, they're walking like I showed you, okay? And what we can do is that we can shoot a laser beam inside the cell or after taking these objects out of the cell. And so imagine this is that spherical bead that I showed you and it is being carried by a motor. And I shoot a laser beam and I focus the laser beam on this particle, and this laser beam acts now like a spring, okay? So you can see that. So as soon as the laser starts, this slows down and this motor tries to pull this bead out of the center of the trap. And you have to imagine something like this happening. Here is the laser beam and it is focused here on that bacteria, okay, let's say. And this motor is trying to pull it out, and this is what, this is a real experiment where you can see one such bead, it will move out there. Tries to kick out. So you have to imagine a motor which you can't see in this picture. It's trying to pull it out and it falls back. And this pulling out and fall back happens because this laser beam acts like a spring, okay? And we can actually, by doing so, because we know how strong this spring is, how stiff this spring is, we can find out how strong this motor is. Because if the motor can stretch the spring further, then the motor is strong, okay? So by using a calibrated spring, we can find out how strong these motors are. And if there is a motor of this type or that type, then the spring would deviate in different directions. So we can actually measure the number of active motors on in these situations. Okay, and this entire setup that I told you about, I won't go into details of this, is called an optical tweezer. Okay, it's an optical trap or optical tweezer. By doing such experiments, what we were able to show many years ago, and I'm not going into too much details right now, that the particular motor called dynein works very well in a team. Okay, and so if you take n number of dyneins, their forces can add up, but that is not true for kinesin. Remember, that is the motor going in the other direction, and the kinesin motor, if you take an individual motor, which is much stronger than the dynein motors, which exert a much smaller force. But although these dyneins exert a smaller force, they can team up very nicely, and then a team of dyneins, weaker dyneins, can match a stronger dynein. And this is perhaps one way in which the cell controls how, relative activity, dynein versus kinesin. Remember, that is required to take you to the periphery of the cell or to the center, is controlled. Okay. Later, what we were also able to show that these dynein motors assemble on lipid raft domains. These are basically regions of high cholesterol on the membrane of this bacteria or phagosome that has been ingested into macrophage cells. So cholesterol may help in accumulating dynein in large teams so that this bacteria is taken to the lysosome. So cholesterol is not all bad. Okay, cholesterol is a very useful molecule. It's just too much of anything is bad. So that's what. Okay, so coming quickly to the last part, another branch of research in my lab is where we look inside the liver, what's happening inside the liver, and why are we worried about the liver? We are now, what you see here in this picture, this scale bar is about 30 microns here. Okay, I'm sorry, I forgot to put the number here. These are individual cells inside your liver. These are called hepatocytes. Okay, and these cells, you will see inside these cells. So this is the boundary of a cell shown in red. This star is the nucleus of that cell, and here are these fluorescent particles, which are actually little balls of fat. So all this stuff that you eat can get converted inside your body into these little balls of fat, which will go and accumulate in all kinds of cells. You need this fat because it's a source of energy, but as I said, having too much of it is also a problem. Now, these little balls of fat shown here in these fluorescent specks, you have to imagine that here is that little ball of fat, this one, and these are again carried by the same kinds of motor proteins. Kinesin here, it's an old, you must be familiar with this now. It is carried along microtubules, and this, what we were able to understand is this association of kinesin with these particles of fat decides how much fat there will be in your blood. Okay, and what we are trying to do right now is that can we control the binding of kinesin or the activity of kinesin on these fat particles inside the liver, so that perhaps we can help patients with fatty liver, obesity, or diabetes. Okay, so this is a very, we believe that nobody had known about this, and we figured it out that this kind of a mechanism happens inside the liver, which controls how much fat is sent out from the liver. Okay. Last few minutes, another very important question in this respect is, if you look at a cell, okay, so here is a cell inside the liver. Okay, now you are seeing only one kind of particle, these lipid droplets. But there are many other kinds of things inside the cell. There are mitochondria, there are endosome, there is a nucleus, there is endoplasmic reticulum. Each of these is a little factory, okay? And these factories need to interact with each other, talk to each other for the cell to function. And that's an example here. For example, this is the nucleus of the cell. This is the boundary of the cell here. And here are all the different kinds of things that exist inside the cell. And they need to talk to each other, and for that to happen, the membrane, okay, so everything is bound by a lipid membrane, as you know. The membrane of this organelle has to fuse transiently with the membrane of the endoplasmic reticulum or of other kinds of organelles. And we have what we were able to do. I won't go into the details of this. We were able to reconstitute such membrane fusion in a cover slip. Okay. And when we did that, we, this is a very useful assay to understand how membranes fuse with each other. And fusion of membranes is a very important target for many kinds of drugs. And I won't go into details of this. I think I will stop here right now. I'm almost at 20-25 minutes. And I would like to thank all of you. And I will go of this mode so that I can see people. And I'm at IIT Bombay, and this is a very nice view from our rooftop. And if you are here, please do visit us. Okay, thank you.
Thank you, sir. The session is open for discussion. Any questions?
Yeah, Professor Rup Malik, thank you. Thank you for a very interesting presentation. This is Vipin.
Thank you.
Yeah. Professor, when we see your talk, what it reminds me is about this microcosm, macrocosm analogy that which we see in scientific literatures and philosophies. What we observe in real life is actually a parody of the same thing is happening in the subcellular level. And the way in which you have presented it is, it beautifully reminds probably all of us who are sitting here in this conference, it beautifully reminds them. Thanks for a very interesting talk. Thank you.
Anybody wants to ask any question?
Okay, we can move on then.
Okay, I think there is no question.
Sure.
Thank you so much, sir,
Thank
for
you.
delivering such a informative talk. You have explained the importance of most.
Unshare my screen. Hold on a minute. Yeah. Okay. Yeah. Okay.
Yes. The motion of dynein and kinin as tug of war, which has helped us to understand the concept in a better way. The motions and the animations have helped us to relate to your talk. So we generally think that our limbs, that is hands, are responsible for our motion. But today, sir's talk has really given us a new angle to look at a real mastermind of motion. So thank you, sir, for taking us to a deeper version of motion. Thank you, sir.
Thank you.
Now our next
would hand over-
invited speaker is. Now our next invited speaker is Mr. Darshit Patel, sir. I request Head of the Department, Zoology Department, Dr. Sarode Madam, to introduce our invited speaker, Mr. Darshit Patel, sir. Over to you, Madam.
Good afternoon. Respected dignitaries, delegates, and student friends. It is my great pleasure to introduce Mr. Darshit Patel, sir, who is co-founder, Chief Scientific Officer of Decode Age, Bangalore. Sir has completed graduation in chemistry and biotechnology from Ferguson College, Pune. Sir is master's in drug design and biomedical sciences and recipient of gold medal of Bharati Vidyapit, Pune. Sir has been worked as visiting faculty member and associate research of Bharati Vidyapit. On his credit, there are many national and international papers having high impact factor. Sir is author of books of reputed publishers like Taylor and Francis. Sir has worked on a research project, metagenomic profiling of bacterial fingerprints of freshwater bodies of an urban location of India. Sir is entrepreneur, researcher, futurist, and devote helping people and decode longevity equations. Sir has also trained many young scientific professionals in his field. His work is focused on slowing down the aging process, decoding the health biomarkers, and creating community awareness. I request Mr. Darshit Patel, sir, to deliver the talk.
Thank you so much, Dr. Sarvade, for that kind introduction. I'm very, very humbled to today to speak among such great professors that I got to listen to today from IITs and many other great dignitaries that are present here. If you'll allow me, I'll just share my screen. Right, is my screen visible and am I audible? If someone can come
Yes,
on that.
yes, sir. You are audible and screen is also visible.
That is great. Thank you so much. So today I want to talk about something that we really find very commonly in the science, and most of us believe that there was two events. Like one was the microscopic event and one was the macroscopic event that eventually framed human beings to where we are. And one of the first theories, that is the microscopic event, is when a biological entity couldn't find an energy source. It made a collaboration with another organism that had its own genome and was responsible for making energy with it. What we call mitochondria today, basically. We see that that's the only cell organelle that has the genome of itself within our cell, apart from the nucleus. So that kind of collaboration gave us the boon of energy. But then the second event that happened at a macroscopic level, once the organism formed, was that of the microbiome. We know now that our body is harbored by literally many microorganisms, be it bacteria, virus, fungi, or archaea. We have them in the respiratory tract, we have it in our gut, we have it in our kidneys and even brains these days. So we are all aware about the microbiome. But we were very much limited until the year 2000 because of the sequencing technologies and mostly the expenses for the same. This is why we couldn't really deep dive into what was happening. Well, thanks to metagenomics, more studies are being carried out in microbiome now, and already a lot of associations have been established with health and disease conditions. But what's the latest moonshot? It's about aging. We are following aging as now a biological condition, because that is probably the only biological condition that is common to each and every individual that is born. Unlike the diseases are found in cohorts of patients. Aging is something that has been central to each and every human being that is present on the planet. So scientists today are looking at aging as a way of intervention, as a way of slowing it down, prevention of chronic diseases, and gut microbiome could be one of the big deals in this particular sector. And that's what I'll be like to talk about today, and some work that we need to be at Decode Age to find out the connection between two. So first, to do the very basics, I'll finish this off in a while. Because we see our gut microbiome has been decoded over many projects. One of the most important one was the one in America, the Human Microbiome Project. And we now have at least certain kind of understanding about what our basic microbiota looks like. On a general term, on a phyla basis, on a kingdom basis rather, we have 96% bacteria, which is not at all surprising considering the organisms adaptively. And then we have fungi and archaea, which usually takes up the rest. Relationship between here, because a lot of antimicrobial peptides that are formed by bacteria are actually used to control opportunistic fungi like Candida albicans that can cause lethal infection. But the probiotics or the commensals from our bacterial group, the opportunistic fungi and viruses are managed efficiently. There is even a recent research into how bacteriophages are targeted by the bacterial metabolites, but that is a topic for another day. Well, if we focus just on the bacteria, which is obviously the 96%, and today we know more about bacteria because the 16S technology that is being used to decode the bacterial microbiota of the human organism through stool samples is very cheaply available these days. For the other kind of like the virus, fungi, and archaea kingdoms, we really need to go deeper. We need to utilize different pipelines, and I think more and more research will come across that side. But for now, if we focus on bacteria itself, so if we divide the whole gut into the stomach, the intestine, which is the small, and the large, and finally the colon. So you'll see a specific kind of bacterial population in each of these sectors of your gut. For example, in the stomach, the CFUs that is usually used to measure the colony forming unit, which is the unit for measuring growth of bacteria. So we'll see lactobacilli, streptococci, and yeast often found in the stomach area, where the environment is slightly acidic. A lot of fungi grows there to digest the food. The lactobacilli is usually used to control the pH as well. If we move down to the intestinal part, which is where the major metabolism, absorption, and reabsorption of food and water happens, is where you'll find the maximum diversity of bacteria. If you look at jejunum and ileum, you'll see a lot of lactobacilli, again, a great probiotic. If we see a lot of our relative abundance into the gut is focused by lactobacilli. Then there is streptococci. The Bifidobacteria is often found in rare amounts, rare percentages, usually less than 2 to 3 percentages. But even at that concentration, they're very useful in controlling inflammatory process and gut permeability because of the bacterial metabolites they are able to release. On the other side, if we come to colon, there's a large diversity there. We'll see more bacteroidetes and Bifidobacteria compared to lactobacilli. So we see that there is a late metabolism stage at this particular part of the gut. Along with that, you will see a lot of, opportunistic bacteria there. So the day your colon microbiome gets disrupted, there is a chance for all these infectious bacteria to grow more and cause certain kind of a health effect or disease pathology into your gut. And we'll see that because we see a lot of connections with the diseases as well. Now, mostly talking about if we try to limit an organ to itself, we are making a big mistake out here. Because when the problem occurs in the gut microbiome, it is often transported through various places, like the brain. There is a gut-brain axis, which is accessed through the vagus nerve of your body. Then there is a gut-liver axis through the portal vein that is controlled. Then there is a gut-heart axis, which often flows through the gut-liver axis. And there is a gut-skin axis directly responsible for sebum production. Why are these important is because even today, like for example, taking consideration into brain, right? So we need a lot of neurotransmitters like dopamine and the serotonin. And we have seen recently that most of the production of these neurotransmitters is now taken care by your gut microbiome, which is then eventually transported to the brain through vagus nerve. So if we see the GABA receptors, the 5-HT, the short chain fatty acids like the acetates and the butyrates and the propionates are very important in maintaining the entire homeostasis between what is happening in brain and what is happening in gut. And recent correlations, you will also see that Alzheimer's and Parkinson's could be predicted early just by the status of your gut microbiome. So this kind of discoveries is really going to change our outlook from a diagnostic perspective to a predictive perspective, because then we can find out early what's going to happen just based on the prediction of what is happening in your gut microbiome. Now, on the other side, if we talk about liver, secondary bile acids are very important. If we look at the elder generations, that is the research that has happened on the blue zones, where people are living at an average lifespan of 95 and above. There are about 5 blue zones in the world. And at this age, in these areas, people have found that the bacteria that are responsible for producing secondary bile acids are often being found in a more relative abundance compared to the younger generation. So at that age, at extreme ages like 95 and above, if you want to maintain health, a direct correlation has been established between how secondary bile acids produced by gut microbiome preserves the homeostasis. When that happens, the TMAO production from the liver to heart also reduces, which causes less atherosclerosis conditions. So this entire network of what's happening through the secondary gut bacterial metabolites of the gut microbiome traveling to various organs and what's happening there. So that entire network has come down to a great limelight in the recent literature as well. And one factor that about gut that controls everything is the gut permeability. Our inner intestinal linings, if they rupture, what happens is the same microbiome, which is responsible for carrying out good effects in your gut, will now enter your bloodstream and translocate to a different organ where it will disrupt the gut microbiome. So the organ microbiome. So your gut dysbiosis is directly responsible for what is going to happen in other organs as well. And we have just described 4 here, but there are many other, like a gut adipose excess, there is a gut kidney excess, and there are a lot many others. And we see that how do we predict diseases? And there has been proper cohort-based research which specifies that growth in a particular bacteria can be correlated to colorectal cancer. And they have eventually found out the mechanisms of that happening as well, providing a pathway from correlation to actually causation. So when we talk about correlation, we see that between gut microbiome and colorectal cancer, there is an increased growth of Fragilis, E. coli, Enterococcus faecalis, and Streptococcus galolyticus. So this is the correlation that was established about which species were overgrown or undergrown in patients that had colorectal cancer. On the other side, when we checked out the causation, they found out that there were 2 cohorts, one in Iran and one in Finland, and it was noticed that higher abundance of certain rare species like Herbispirillum, Cannatibacterium, and Barnezella was directly associated with patients experiencing DNA damage. Now, not just DNA, that is a direct cause that it is causing DNA damage. And they eventually found out through which mutations. So TP53, RAS, and NRAS, which are the common tumor-based mutations, were eventually established. That shows a direct correlation between how gut microbiome can cause CRC. The idea behind what we are doing at Decourage to produce patient reports of gut microbiome prediction is to understand about at what levels these species can grow and turn into dangerous ones causing DNA damage. Not just that, the secondary metabolite production mechanisms have also been identified, like the N-nitroso compounds and the hydrogen sulfide production. So if we see H2S, which is the hydrogen sulfide production, the gas producing genes in the bacteria are now directly correlated to CRC formation as well. So if you take one of the recent CRC early diagnostic test or early prediction test, they test the production of hydrogen sulfide from your gut microbiome. So that is an important breakthrough that has helped in early detection of CRC. Now, finally, what we are looking at in the next stage is genotoxins that are released, like certain bacteria like PK positive E. coli, right? Such bacteria would produce certain genotoxins, which eventually lead to DNA damage and responsible for tumor suppression, responsible for tumor formation. Such some of the few examples are cyclomodulin cycle inhibiting factor and cytotoxic necrotizing factor 1, which actually stops the apoptosis. And when apoptosis is stopped, that means the cell does not die and keeps dividing, which is the other definition for tumor. So that's why such genotoxins that can directly affect apoptosis has been found to be an important correlation between what is done by gut microbiome to actually cause the colorectal cancer. If we come down from tumor to metabolic disorders as well. Now, type 2 diabetes is one of the most explored disease when it comes to correlationship with gut microbiota. And we have seen a patient cohort of around 2,169 patients, and they showed that people with higher insulin resistance have lower Shannon index, which is actually the amount of species, the number of species that are there. And the species richness is overall low. So it has been repeatedly established in different kinds of research papers, and even we have found out that for a healthy microbiota, species richness and diversity is very, very important. Because the lower number of species, that means lower homeostasis, a higher number of species means higher homeostasis. So that has been the main correlation. Again, we come down to Formic population that is often found to increase along with the gram-negative bacteria, and there was less diversity and stability. Less diversity is the core problem in any metabolic disorder that has been associated with gut microbiome. We will still see certain species coming up which has higher abundance, which means their higher abundance is directly associated statistically to the T2D patients. And you see some of these like Christiansella, then we have Marvin Bryanta and Ruminococci, which was often associated with lower IR. Lower IR, better, you will not get diabetic soon enough. But then if you have a higher insulin resistance, that means there is a problem there. And these bacteria fortunately were all known to produce something called butyrate, something that we saw in the gut-brain axis as well, something that comes across in gut-liver axis as well. So short-chain fatty acids like the butyrates and the acetates and the propionates are very important when it comes to the gut-brain vagus correlation and even maintaining the gut intestinal health, which we talked about. If we all know, like if we follow the Indian tradition, we say that colostrum is very important to maintain our health. And that is because it maintains the intestinal lining, which actually allows the bacteria to stay within the gut and not translocate to some other organ to cause a metabolic disorder. So to sum it up all, between the gut microbiome and aging, we see that there is a reduced microbial diversity, a higher number of pathobionts that come across, the gut motility, the movement in the gut reduces, and the gut permeability is high. That means the intestinal lines are very much damaged. There is a higher inflammation because as soon as there is a damage, there is an inflammatory response. Plus the bacterial translocation from one place to the other causes an inflammation in that particular organ as it is not used to the gut microbiome. We have lower metabolism and increased disease predisposition. There is a cohort of around 40 diseases now that can purely be predicted based on your gut microbiome. And this could be anything autoimmune disorders as well, like the Crohn's disease and the ulcerative colitis, which is one of the most talked about topics in India these days because of the number of patients, obviously. And on the other side, the gut will eventually give you higher infections, gut disorders, even Alzheimer's and Parkinson's, like we have always talked about. Then hypertension, dementia, and stress. So for hypertension, especially blood pressure, there has been a direct correlation between SCFA production, GPLR protein activation and inactivation, and therefore causing blood pressure going high. So this is the overall summary. And if we now purely move to hallmarks of aging. So just like in the year 2000, we had the first hallmarks of cancer, which really helped us accelerate our research towards the cancer therapies. We had the hallmarks of aging because now the WHO considers aging as a disease. So they had to establish an hallmarks of aging, which in 2011 was around 9 of them, which include everything from genomic instability, the loss of proteases, there were epigenomic alteration, telomere attrition, obviously the Nobel Prize winner, Dr. Elizabeth Blackburn. And then we have cellular senescence, stem cell exhaustion, and altered intercellular communication. These were the first 9. But in 2022, in a conference in Copenhagen, 5 more hallmarks were proposed, and one of them actually was the microbiome disturbance. The question here is it right to include the microbiome disturbance as a hallmark of aging? Because what we have come across repeatedly in the research is that microbiome is actually one of the leading factors or a cause to the rest of the 9 hallmarks of aging. Like we saw in the example of the colorectal cancer, there were genotoxins that was actually damaging the DNA, causing genomic instability, which is a hallmark of aging. In terms of T2D, we see insulin resistance pathways being affected. So that there is a problem there. There's a metabolic disturbance there. There is a kind of deregulated nutrient sensing which is happening there, which is another hallmark of aging. And on the other side, we have also seen that microbiomes can lead to cellular senescence because of release of molecules that mimic SASP, which is the senescence-associated secretory phenotype. So that is a direct correlation to contribution into the cellular senescence hallmark as well. Similarly, if we are able to find the connection between how microbiome and microbiome metabolites are contributing to the rest 13 hallmarks of aging, we can actually move that particular hallmark into the primary stage and then say that it is the cause of rest of the hallmarks of aging. And that would give us a much more clearer idea apart from creating a confusion of multiple hallmarks of aging. So that is a theory that we work on in Decode as well. Now, when we say that gut microbiome can be used to control aging, it is really important. Sorry, I'm not out of my time, right? I'm just confirming that.
Yes, sir, you can continue.
All right, thank you. Right. So if we look at some of the centenarian studies that have happened, so centenarians, people living 100 and above. So in Italy, there's an area called Sardinia. We see certain specific things. There was no change between the age 30 to 70, but as soon as a person crossed 70, and it was compared to the younger ones, we saw much lower species diversity in the higher age people, especially the centenarians. We saw a lot of opportunistic infective bacteria like proteobacteria, lower SCFA, lower saccharolytic potential, which is the ability to break down sugar to produce energy, and higher proteolytic function, which is cutting down of protein more. Similarly, in Ireland study, sorry for the typo there, it's Ireland study, they saw that there was a higher bacteroidetes and Clostridium levels, which is Clostridium difficile is one of the biggest infections and cause of many gut disorders in humans right now. And we see a lot of new techniques that are being used to deal with it. And in long-lived healthy individuals, there was a higher Akkermansia. Now, this name is very important and is coming across repeatedly again in the past 3 to 4 years, that Akkermansia is one of the most important rare species that can be utilized as a therapeutic intervention. And now the most important global moonshot is that the gut aging clock has to be developed. It's to understand what's your biological status based on your gut, such that new therapies can be used to intervene and prevent the gut dysbiosis and related disorders. Finally, if we move on to what are the applications. So there are 3 main applications that are currently being worked on. One is microbiota-based interventions. We have all heard about prebiotics, probiotics, but then there is something upcoming called the life culture therapies, which is literally planting live bacteria into your guts to help them grow at the optimum way. Then FMT, one of the most interesting therapies that have come across recently, the fecal microbiota transplant, which is taking something from a younger or a healthier person and putting it into an older person, a fecal matter, such that it helps growth of good bacteria into the diseased person. But a recent study on rat proved that taking the gut microbiome of the younger rat and putting it into an older rat actually rejuvenated the metabolic processes that were happening in the older rat. So such things, if they can be taken to the human clinical trials, it would be probably one of the most important breakthroughs in terms of preventive treatments to come about. And dietary interventions would get more data-based and it would have more data points to be worked on rather than some pseudoscience based on just observations. So these are the 3 main interventions that we can think of and which we are trying to provide to people. So at Gut Microbiome Research at Decodage, first of all, our challenge was to understand what is the best technology we can use to understand the gut microbiome. Because there are 2 currently used, which is the 16S and the whole metagenome. So in 16S, we had a real big limitation. We could go to the bacterial level, right? But only bacterial level. If we had to try out the fungi and the archaeal pathway, we had to go for different kind of amplification methods and different pipelines like 18S and ITR. But with shotgun, things got very interesting. Because 16S is just amplifying a gene and giving me a taxonomy, shotgun is just that pure pool of DNA that I've got from the stool. We could break it down properly. We could understand what are the virus, what are the kind of fungi present, what are the archaea, and not just that. With shotgun, I got a greater depth into my data, and we actually started finding and predicting what are the kind of functions that particular microbiome can perform. And we got a list of around many secondary metabolite gene producing genes in the bacteria and in the archaea and fungi as well. So that was a great interesting study which we have just completed. Unfortunately, I cannot reveal the data, it is yet to be published. And then we had to work on pipelines and automations of functional prediction and associations. Because for associations, we need more and more data. Currently the data, especially the Indian microbiome data, is at dearth. We'll hardly see 7,000 to 8,000 public samples, and that's why we had to start our own sample collection. Our idea is to curate a database to find the features of an aging gut microbiome. What are the functional changes? What are the overexpression of genes? And what are the different dysbiotic microbial populations that we get? And finally, we are doing a centenarian study for India as well. So just to quickly brief what we have found out so far, So when it comes to 16S and metagenomic or the shotgun comparison that we did, so one of the main things that we found is like functional profiling becomes very easier with shotgun with higher amount of data, but the reference databases are still not so optimized. So building that is more important. With 16S, you can get to the functions, but it will still be deduced from the taxonomy rather than found out directly. With the taxonomic resolution, we can get to the strain level with shotgun. With the 16S, it has been difficult to go past species level, but with shotgun, it has become very easy to go at strain level. And that really gives us depth into what we have analyzed. Taxonomic coverage, like we have already discussed, that with shotgun, we can find all the different kingdoms, whereas with 16S, we'll have to use different primers for ITS and 18S to find out the archaea and the fungal species. Now, databases in shotgun are relatively new when it comes to gut microbiome specificity, because often using a generalized database has given a lot of false positives in our data, and which is what we want to overcome with curating better databases. Now, sensitivity to host contamination is obviously more when it comes to shotgun, because there is no biasness in selecting and we are not amplifying any particular part of the genome, which we do in 16S, and that's why we fall better with the sensitivity of whole genome. The bias is obviously higher in 16S for the same reason, because we are amplifying the genes. In shotgun, it is less because we are going for a total DNA sequencing of whatever we have got. Now, finally, we compared some healthy and disease samples specifically type 2 diabetes. We saw certain key findings that the healthier gut sample was more diverse, which we have discussed again in the previous slides. It is necessarily to have a higher diversity to maintain a better health. Specific type 2 diabetes associated microbes were also identified in our research, which correlated with all the regular research findings that was found throughout the world. And that probiotic strains were more abundant when it came to the healthy patient compared to the type 2 diabetes patients. The future that we are thinking of based on the research as we completed is to find out preventive interventions based on gut microbial composition. And our major observation was that environmental nutrients and heavy metal association studies can be used to better understand taxonomy of gut microbiome. Because what we are having is contributing to the 70% of the gut microbiome, what we eat. So it is really important to take that as a metadata when we go for more and more samples. And finally, we have an ongoing study, which we would love all of your help to. We are identifying people who are living at 100 years and above in India currently, and we are collecting their samples. Successfully, we have completed around 15 of them, and that analysis will be presented in the next conference, which is coming up in December. And our objective here is to find unique signatures that allowed Indian centenarians to reach the stage that they are, and then compare it with younger cohorts to find out the real meaning of biological aging at extreme lifespan. So thank you so much, GE Society, for allowing me to present our ideas and work at your repeated conference. I'm very glad to have heard the previous talks as well from wonderful Dr. Roop Malik and Dr. Vibin. And yes, that is our current team at Decourage, and I'm open for questioning actually.
Thank you very much, sir. There are some questions in the chat box.
Right.
So the first question is how to increase gut microbiomes, and is there any relation between gut microbiomes and diet containing millet food?
Right. So to address the first question, rather than asking how to increase the gut microbiome, we must look how to balance the gut microbiome. For example, the food that we are eating is predetermining what gut microbiome is developing. For example, if you see a very interesting study, if you see vegetarians, you will see higher Parvotella, which is the fiber degrading. If you see non-vegetarians, there'll be higher Bacteroidetes, which are mostly meat degrading enzymes. So based on what you are eating, it is really important that you develop the right probiotics, and the balance It is not about increasing, but creating the balance between the good and the bad bacteria. And that balance will come with the kind of food that you eat and the kind of environment you live in. So we often hear in the internet that we should have a plate full of 7 colors. And those 7 colors are basically substrate to different kinds of microorganisms. The more colors, that means the more material to degrade. And when there is more material to degrade, there are different kinds of bacteria that are growing on that substrate. So that becomes an important part. And of course, through maintaining a good diet, it is really important to do that because that will help you develop a good gut microbiome. If we talk about probiotics that are currently being sold, it is not entirely a good option because it has a prefix CFUs of different kind of microorganisms. Right? Maybe you have the enough lactobacilli in you. Then why are you taking it in the supplementation form? So we need to go more personalized when we come to supplements. But if we are talking about how to balance it, it is purely diet.
Okay, sir. So there's one more question.
Sure.
Sir, can you please explain about genotoxins and how it is produced by microorganism and how it is related to aging?
Right. So absolutely. So first of all, we see that when bacteria are not given the right substrate, they will not make the right metabolites. It's as simple as that. So if you give the right substrate, that is a complex carbohydrate, to your bifidobacteria, it will metabolize it to make something called short chain fatty acid, which is eventually going to help you because it is an anti-inflammatory. But if there is not the right substrate, maybe you decided to have some junk food, and maybe there was not a right molecule there to be degraded. When the gut microbiome or a particular species in gut microbiome degrades that bad substrate, it will produce a toxin. Now, what is this toxin going to do? This toxin might enter your bloodstream, go to some other organ, and it is going to create a damage there. And one of the most important kind of damage is the DNA damage because it can cause a double-stranded break. For example, colibactin, which is found in CRC patients, will see that there is a double-stranded break in their DNA. And that is actually caused by colibactin, which is released by EK positive Escherichia coli. So that entire mechanism that happens is eventually going to cause you disease. And how DNA damage is going to cause aging is a question of hallmarks of aging. Something has been established that when there is enough genomic damage accumulated in genome of your cells, it is going to start behaving in a way that is going to contribute to faster aging. So this entire mechanism starts at a bad substrate, a bad metabolite, which is a toxin. That toxin goes and create the DNA damage, and that DNA damage is going to eventually contribute to the aging process. So that entire mechanism flows through.
Okay, thank you, sir. I request Dr. Sarode Madam to give vote of thanks.
Thank you, sir, for sharing your valuable knowledge with us. Sir has explained very well about gut microbes, composition of gut microbes, how these gut microbes are associated with diseases. Sir has also told about gut microbes as a hallmark of aging. Application of various gut microbes in aging research and various technological challenges and future on how to study gut microbes for human health. Sir, definitely it enhances our knowledge and about the role of gut microbes. Thank you, sir, for very nice informative talk. Thank you.
Thank you so much. Thank you.
Thank you, madam. Thank you, sir. Our next speaker for today is Professor Park from South Korea. I request Dr. Kirti Patel, sir, to introduce our speaker. Over to you, Kirti Patel, sir.
Good afternoon, sir. Good afternoon to one and all delegates, participants, and my dear friends. It is my pleasure to introduce today's guest speaker. But first, I would like to thank my dear friend, Dr. Deepak Borste, who introduced us, Professor Park. Professor Park has received bachelor's degree from Honjik University and PhD from Pohang University of Science and Technology, Republic of South Korea. He has joined as postdoctoral fellow at Center for Smart Supramolecules, and later at Harvard University from 2008 to 2012. Sir then worked as senior researcher at Samsung Electronics and as adjunct professor at University of Science and Technology, Republic of South Korea, from 2015 to 2018. In between, he has also worked as team leader and group leader at Institute of Basic Science. Currently, he is assistant professor at Department of Biochemistry, Daegu Catholic University, Republic of South Korea. His research interests includes biomaterials, chemical biology, bioimaging, and proteomics. He has published more than 50 publications in reputed international journals, which include some of them in Nature Communications, Angwante Chemie, Journal of American Chemical Society, Accounts of Chemical Research, etc. He has also contributed in 2 books and has 6 national and 6 international patents on his name. Uh, Without taking much of your time, I request Professor Park to take over the session and deliver his talk. Professor Park, we welcome you.
Okay, thank you very much for your kind introductions. And also, first of all, I would like to thank the organizing committees, right, for having me here to participate in this very special conferences such as speakers. So, although it's a little unfortunate to not give a talk on site, but through online, but still, it's a great pleasure for me to present my research in this very special occasions. Thank you again for the invitations. So when I first got the invitations of this talk, I realized that the theme of these conferences here, such as the recent advances in chemical and biological sciences, so matches well with my research topics. Technically, I'm a kind of like the supramolecular, especially for the supramolecular material chemist. Sounds a bit far from the biological sciences, but as a supramolecular material chemist, so I have developed a number of tools that are useful for the biology and even the biomedicine researches. So I believe today is a good chance to let you know how the supramolecular chemist like me helps the biologists and the biomedical engineers. I hope you may enjoy my talk today. All right, so let's get started. Okay, so I think let me start from the very general introductions. So I think that everyone here might know what this is, right? As shown in here, yes, it is a glue for, especially for the multi-purposes. It's very useful in our daily life, right? For repairing the broken part, like cups and toys, and attaching something to the other, like a hook on a wall without any damages, right? And like this, ladies, I think ladies knows it much better than I do for their beauties, right? Wearing a fake eyelashes with help of this gluing, right? We are using glue quite often, so because simply it's easy and convenient to use, and of course it's cheap. Okay, so we are using the glue mostly for the 2 reasons. One is for repairing, applying a glue on the damaged part and adhere them together, right? And the other is attaching a function to other materials, like a function for hanging on a wall and a function for your beauty on your face, right? So you can glue such materials in daily life whenever and wherever you want. So because it's all in a manageable scale with your hands, right? But even a very tiny things like this, so you can still handle it. So you can grab it with a tweezers and apply glue on it, then place it whenever you want to attach. However, if you think about the really small world, such as micron scale regimes, that you are now in trouble, right? You cannot even see it. So as you can simply imagine, the gluing is not easy job anymore in that case. So when it is going down to a cell levels, so it's even more difficult, right? because of not only a small size, but also the interfering from the other small molecules such as protein, carbohydrate, and lipid, which are all dynamically interact with each other in a very complex environment such as cytoplasm, inside the cells. So you probably don't want to glue them all together, right? Then the questions can be, right, how can you glue very small thing to a desired locations here, or the molecules, to add in combine molecular functions in a very complex and dynamic environment. So the researchers actually already suggest a tool, like a protein and the ligand interaction-based high affinity binding pairs, such as streptavidin and biotin. So streptavidin is a tetrameric protein that has exceptionally high binding affinities to its ligand molecules, such as biotins. So quite specifically and strongly, right? Then the binding constant between them is almost reached to the 10 to 15 for mole, which means that one recognizes the other very efficiently, even less than a picomolar concentration. So utilizing such a highly strong interactions, highly strong interactions, this binding system has been used as a versatile molecular glue. So it provides a combined function for various useful applications, including immunoassays, even the bioimaging and diagnosis, and the conjugations of the biomolecule to each other, even affinity-based chromatographies, right? As you can see this graph here, so it's about a number of the publication and patent that are dramatically increased since 1980s. Interestingly, it's mostly related with the biological studies applications, so meaning that streptavidin and biotin, the binding pair, has become a really powerful tool in biology and biotechnologies these days. However, right, it's true that it's beneficial features. It has beneficial features. So as a versatile molecular glue, however, at the same time, it has unavoidable demerits, right? Because it's actually the protein-based system. They cannot be modified easily. Thus, it is difficult to control their binding affinities. And it can be denatured under organic solvents and affected by some of the biosystem, especially by the endogenously existing biotin or already existed the biotin-related protein in the systems. That sometimes the severely reduces the binding ability of the streptavidin. In addition, it's actually expensive since it cannot be synthesized in a large scale, but extracted from the nature in a small scales. Although it has such demerits so far, so there has been almost no alternative system or tool and techniques in biotechnologies as a molecular glue because of its versatilities with high selectivity and strong interactions. So now it's really obvious that if you solve the problem, right, so you can develop the much better molecular glues for advanced biologies and the biotechnologies. So to address the issues here, good suggestions can be the replacement of it with a new high affinity binding system, so that has such merit, right? High binding affinity and selectivities, chemical tractabilities, controllable binding affinity, of course, needed to be small and orthogonal to the biosystem, and we can make it in a large scales. So is there any system like this, right? Or can we really provide such systems mimicking streptavidin biotins, or even better? So the answer is yes, you can actually. So there is a system based on a pumpkin-shaped host molecule known to be cucurbituril. A cyclized the compound made of the glycol to have a cavity like this. The name of the compound is somewhat difficult to pronounce, right? Cucurbituril. So because the name actually comes from the academic name of the pumpkin, cucurbitaceae. It's still quite complex and difficult to say. So we just simply call it the CB. Actually, I have been working on the chemistry of this CCBs for more than 10 years. So especially I'm quite interested in the CB7 made of the 7 pieces of the glycolium because it had exceptionally high binding affinities to its selected guest molecule here, right? Such as the streptavidin, such as the ferrocene or adamantane derivatives with the binding constant ranging from 10 to 12 to the 15 per mole. As you can recall the number, this is somewhat similar or even higher than that of the streptavidin and bioteins, right? And what interesting is the size is much smaller than the streptavidin. And this is a fully synthetic system. The interaction is not interfered with any other biosystems. And also this is the fully synthetic system. We can modify using the lutein, the organic synthetic method, and then we can make it in even large scales. So now you probably agree with me, right? It can be a really good alternative to replace the streptavidin biotin system to serve as the molecular, the glue. Okay. So those using such advantages over the streptavidin biotin system, I have already demonstrated interesting bioapplication of it, including the bioimaging and imaging isolation, identification, and even the purification of the proteins and the selective cancer imagings, and etc. Due to the time limitation today, sorry for not sharing all the results, but I would like to share some of them regarding the protein imagings for today. So when I first considered that the CB7 based the binding pairs as a useful tool for bioapplication, I asked ourselves, like the first, can the synthetic molecular recognition system binding pairs so recognize each other even in a very complex biological environment, such as inside the live cells, where a lot of biomolecules coexist and dynamically behave, because any other synthetic, the host case affinity systems do not recognize each other under such as biological and physiological conditions due to their weak molecular interaction, especially in water. So I think the host